Hello friends, today we will discuss the cell cycle, important phases and their functions. We will also discuss what is the location of cell cycle, why the root tips are fixed at a specific time for the study of mitosis, what happens in mature cells, how the cell cycle is controlled, what happens if the cell cycle control is failed, and finally we will discuss certain multiple choice questions. Let us deep dive in cell cycle. Friends, have you ever thought how the body of organism develops from a single cell, that is a zygote? The zygote is the first cell which is produced by the fusion of male and female gametes. And this zygote develops into embryo which is present in seed. After germination, it produces root system, shoot system and entire body of the plant. In case of animals, this zygote develops into a whole body. Now, how the new cells are added to the body of the organism? It was firstly explained by Rudolf Virchow in the cell theory. He stated omnicellula e cellula means each cell develops from pre-existing cell. The cell zygote, for example, behaves like as a parental cell. It enters into cell division and produces daughter cells. Daughter cell grows for some time and they becomes parental cell. They enters into cell division and produces more daughter cells. In this way, number of cells are added to the body of organism. This cell division results in formation of body or soma. So such cell divisions are also called as somatic cell divisions, specifically the mitosis. In next video, we will discuss mitosis. Let us define the cell cycle. Cell cycle can be defined as a series of events that takes place sequentially, which results in the division of the cell and duplication of the cell. Cell cycle can also be defined as duplication of cellular content and division of the cell. So cell division is the key phase in cell cycle which results in addition of new cells. So this phase is M phase, cell division phase. M phase enters into next M phase, but there is a long phase in between these two successive M phases. And this phase, which is present in between two successive cell divisions, is called as interphase. Now, in this diagram, we can see that interphase is much more longer. So, for our convenience, we will divide interphase into three different phases, namely the G1 phase, S phase, and G2 phase. Daughter cells firstly enter into G1 phase, which is followed by S phase, G2 phase, and M phase. So this is the correct sequence of phases which occurs in cell cycle. Now let us discuss one by one. The G1 phase is named as a first resting phase because activities in nucleus, the activities of the chromosomes are not visible under microscope. Newly formed chromosomes are actually diffused in this phase and apparently the cell is taking rest. In fact, the cell is growing in its size and this phase is responsible for the growth of the cell. So G1 phase is also called as a growth phase. As this phase occurs after mitosis, it is called as a post-mitotic gap phase. Now here we can see the G1 phase is the longest phase in the interphase. Much of the time in the cell cycle is expended in the G1 phase. The cell is physiologically very active. So newly formed daughter cell, it has to enlarge in size. It needs to synthesize various types of RNAs, namely messenger RNA, ribosomal RNA, tRNA. After synthesis of these RNAs, the cell engages into protein synthesis. The proteins are enzymes, and the enzymes which are required for the growth and metabolism in the daughter cells, they are synthesized here in G1 phase. After sufficient growth, the cell enters in S phase. Proteins which are required for DNA replication are synthesized in G1 phase. And here is one checkpoint which checks the entry of cell from G1 phase to S phase that we will discuss in the cell cycle control. S phase is known as a synthesis phase because the genetic material is synthesized in S phase. or specifically we can say that the DNA is replicated. Each chromosome carries a centromere and it has a short arm and long arm. In S phase, this genetic material is replicated, so two sister chromatids will be produced on the short arm as well as on the side of long arm. Obviously, the genetic material becomes double, but the number of centromeres remains same, and therefore the number of chromosomes remains same. This is one important fact which is asked in various competitive examinations like as set examination and net examinations. After completion of the DNA replication, cell enters into G2 phase. As again, there is negligible activities in the nucleus which are visible under microscope or which are not visible under microscope. So this phase is also called as a resting phase. But in interphase, this is second one. So it is called as second resting phase or gap 2 phase. It occurs just before mitosis. So it is also called as a pre-mitotic phase. The cell is engaged in the synthesis of proteins, specifically the tubulin proteins, which helps in formation of the spindle apparatus during the metaphase. So tubulin proteins are synthesized in G2 phase. Similarly, the cell organelles are replicated, which later on will be divided in daughter cells. After completion of G2 phase, cell enters in M phase. M phase represents mitosis phase. This is actual cell division phase. So in number of diagrams, we can see that M phase is represented as CD phase, cell division phase. The cell division occurs by division of nucleus, which is called as karyokinesis, and division of cytoplasm, which is called as a cytokinesis. In karyokinesis, four phases are there: prophase, metaphase, anaphase, and telophase. We can remember them as PMAT. So this is the correct sequence of phases which occurs in mitosis. After completion of karyokinesis and cytokinesis, the parental cell divides in two daughter cells, and these daughter cells again enters in the next phase, G1 phase. So in this way, the daughter cells, they again enters in the interphase where they grows, enlarges. They may enter into S phase, G2 phase, and again enters into cell division phase. If we compare the time span, we can see that almost 50% of the time span of life cycle is expended in G1 phase. If we consider the entire cell cycle as a cycle of 24 hours, about 11 to 13 hours are required in G1 phase. About 6 hours are engaged in synthesis of DNA material, genetic material. G2 phase is about 2 hours long and the cell division is completed in about 1 to 2 hours. The cell division occurs usually early morning. So this time span is about 7 to 8 a.m. As per the season, this time may change. So in summer season, it may be about 6 to 8 a.m. or in monsoon season and winter season, it may be about 7 to 9 a.m. So this is the reason why the root tips are fixed at a specific time for the study of mitosis. Now, once the mitosis is completed, M phase is completed, that is about after 8 a.m., the cell enters into interphase. It completes G1 phase, S phase, G2 phase, and comes into M phase in early morning, that is the next 7 a.m. So, in between 8 a.m. today to 7 a.m. tomorrow, the cell is in interphase. And we have seen that in interphase, the chromosomes are not visible. So we cannot observe the mitosis during this time span. And therefore, we required to fix the root tips at a specific time, that is during the early morning. Now let us see where actually the cell cycle occurs. Cell cycle occurs in the type of cells which are able to divide and re-divide. Now, such cells are called as meristematic cells. And these cells are present in the meristem. In case of plants, this meristem is present at the apex of the root. So, it is called as root apical meristem. And it is also present at the tip of shoot, which is called as a shoot apical meristem. So, this cell cycle is actively seen at the tip of root and at the tip of shoot. but should consist of the pigments, and so it is comparatively difficult to observe the chromosomal activities in shoot apical meristem. So we use root apical meristem or we use onion root tips. So this is the site of cell division and it is without pigments. Now let us discuss what happens in mature cells. So cell cycle is active in meristematic cell. Now what about the other cells which are matured in the shoot and in the mature leaves? Now these cells which are entering in mature body are the cells at G1 phase. But these cells once get differentiated or specialized, they will never enter into S phase. So their entry in S phase is restricted and therefore they continuously remains in their specific physiological activity. Such cells which permanently remains in physiological activities and never enters into cell cycle, they are said to be present in G0 phase. So mature cells are present in G0 phase. Now let us discuss the control of cell cycle. Cell cycle is controlled by a specific group of enzymes or proteins which are called as cyclin enzymes or cyclin proteins. Cyclin D amount controls the entry of cell from G1 phase to S phase. So this is the first checkpoint. Similarly, the cyclin A and cyclin E controls the entry of cell from S phase to G2 phase. So this is the second checkpoint in the cell cycle. The content of cyclin A and E ensures that the DNA replication is correctly completed. In G2 phase, there is cyclin B which controls the entry. So this is another checkpoint. And in mitosis, there is one complex which is called as anaphase promoting complex, APC. It ensures that all the chromosomes are arranged at the equator and now the chromosomes are ready for the splitting of the centromere. Once it is assured, then this complex triggers the splitting of the centromeres and entry of the cell from metaphase to anaphase. So this is anaphase promoting complex. Now all these cyclins work with the help of cyclin dependent kinases and the amount of CDK remains almost constant during all the cell phases. If there is a fault in DNA replication or the DNA replication is abnormal, then a specific protein is synthesized which is called as a P53 protein. This protein triggers apoptosis of the cell. So such abnormal cells will be digested by themselves. Similarly, in mature cells, one specific protein called as P27 is synthesized. This protein binds to cyclin D and in this way it inhibits entry of G1 phase into S phase and such cells permanently remains in G0 phase. This happens in mature cells. Now the question is same. What will happen if this kind of cell cycle control is failed? Now when control is failed, the cell divides repeatedly at unwanted time, at unwanted place, and in unwanted number. Ultimately, this unwanted growth will result in formation of tumor. This tumor is nothing but a cancer. So failure of cell cycle results in formation of tumorous cancers. That's why the control of cell cycle is very important. Now let us check our understanding. The first question is that dash dash is the longest phase in the cell cycle. DNA replication occurs in which phase? Correct. The proteins required for spindle apparatus are synthesized in dash dash phase. RNA are synthesized in dash dash phase. Now this is one tricky question. The cell is physiologically most active in dash dash phase. And the final question is that cell division occurs in dash dash phase. The questions are very simple. Write your responses in the comments. You may write your responses in very short forms like as G1 phase, S phase, G2 or M. Simply write down the 6 letters in the comment section and let us check your understanding of the cell cycle. If you found this video helpful, don't forget to like this video and subscribe my channel for such informative videos. See you in next video. Thank you.